Hello friends, welcome to yet another intriguing edition of Rahul's Advanced Biology. Today I will be bringing you a very ecstatic topic known as the transcription in prokaryotes, the elongation part. In my previous videos, I have already described at length about the initiation process of the transcription in prokaryotes in the Escherichia coli. Now in this very video, we will be talking about the RNA polymerase dynamics. How does it undergo changes while adding the NTPs, and how does it overcome various challenges posed by this dynamic process. Now, in order to understand and comprehend this process, let's talk about the RNA polymerase movement model. How does the RNA polymerase really move when it binds with the DNA, and how does it cause the formation of the mRNA, how does it cause the addition of the incoming nucleotides, and how does it again break and reassociate with the DNA. So all those dynamic processes will be covered in this very video. Now let's get down to business. First of all, the RNA polymerase movement model. Now the RNA polymerase in E. coli, you can see there are various things written over here. The RNA polymerase contains a groove and the active site mainly. The groove binds with almost 17 base pairs of DNA. The active site of the RNA polymerase core enzyme binds with the 13 base pairs of DNA. Thereby the groove and active site, they cumulatively bind with 30 to 35 base pairs of DNA. That's point 1 to be noticed. The groove of the RNA polymerase core enzyme is lined by positively charged amino acids. Thereby it enables the RNA polymerase to bind to the DNA which contains the phosphate groups and which are negatively charged. The DNA is negatively charged because of the phosphate group PO43-. Therefore, it binds the DNA with affinity, that is known as electrostatic affinity, right? Due to oppositely charged ions. Then the active site is also lined, is also assisted in its conformational change by binding with the cofactor known as the magnesium +2 ion. Right? It's indispensable for the conformational change, the favorable conformational change in the active site of the RNA polymerase. Then the active site also known as the catalytic site, right? How does it get formed? It forms via a cleft. A cleft is like a region where you have some kind of depression for the formation and the attachment of the DNA, and thereby producing and synthesizing new mRNA transcripts. Now, the cleft between the two large subunits of RNA polymerases, it's responsible, it's accountable for grasping the DNA, and that entire structure is known as jaws of RNA polymerase, right? That is also a part of the catalytic site. Now, the length of the RNA hybrid. RNA hybrid means the RNA DNA hybrid. It should not extend beyond 8 to 10 BP. Otherwise, what you will be able to notice is the R-loop formation, which could be detrimental. We will be talking about it later in this very video. Now, the length of the RNA DNA hybrid must not exceed a certain limit, that is 8 to 10 base pairs. So that is put in check by a special protein known as the LIT protein, right? The entry of nucleotides, the entry of the NTPs, ribonucleotides, not the deoxy, the NTPs, and in place of thymine, you will be having uracil in the mRNA or any type of RNA. So the entry of nucleotides into the active site happens how? It happens via a secondary channel present in the RNA polymerase. It's known as spore in yeast or Saccharomyces cerevisiae, right? That's how the entry of the nucleotides occur. Now we talk about the very transcription bubble. It's 8 to 10, mostly 8 to 9 BP of DNA RNA hybrid. That's known as the transcription bubble. If it exceeds or if it is less than that of length, then 8 to 10 BPs of length, then you are in major problem. Now, how does it all happen? How do you really add nucleotides and again you re-add the nucleotides? How does the cycle really keep on continuing ceaselessly? So we have something known as the Brownian Ratchet mechanism. This mechanism is the most accepted mechanism in research, which describes to you that the RNA polymerase must bind with the incoming substrate known as the NTP and also with the template strand of the DNA. And it must form the associations, and then for the next cycle, which means the next incoming nucleotide NTP, it must break the previously made associations or bonds. So thereby, it's known as the Brownian Ratchet mechanism, where the energy which is produced after the binding of the correct NTP, after the release of the phosphate groups, right? the energy that you get, it is responsible for stabilizing the conformation of the RNA polymerase, especially the active site of the RNA polymerase. And then before the NTP binds, before the NTP gets added, the trigger loop in the RNA polymerase unfolds, and after the NTP binds, the trigger loop folds, and this cycle keeps on continuing. Right? This is the most accepted mechanism, the Brownian Ratchet mechanism, where the energy which is driven or which is received after the NTP addition, the correct NTP addition, let me tell you, so that there is no conformational mishappenings occurring. That stabilizes the active conformation of the RNA polymerase, active site, and the entire RNA polymerase itself. And then that trigger loop, it unfolds before the NTP gets added, and after the addition of the NTP, after this very step, it folds back into itself and gets the active conformation in its favorable form. And this cycle repeats and repeats until and unless you find the termination sequences. Now, R loop formation. R loop formation happens when the real research behind this R loop formation, you can see a latest paper. It was released few days earlier. 8 to 9 base pairs of DNA. This is the major part. If the replication bubble, or if the transcription bubble increases or it crosses the 8 to 9 or 8 to 10 base pair of length, it comes around 12 to 13. What happens is R loop formation happens. R loop is nothing but you have loop in the nascent mRNA, a triple stranded structure where you have loops in the nascent mRNA, along with it binds with the template strand, and you leave the non-template DNA strand. So that's a pretty detrimental state. So you don't want R loop formation if you want the genome integrity to be completely stable. Then we come to the stalled RNA polymerase. What if the DNA is damaged? What if the DNA has some kind of mutations? Then sometimes the blockade in the RNA polymerase traversal occurs due to DNA damage. That blockade may lead to misalignment of 3' mRNA along with the active site of the RNA polymerase enzyme. So the 3' OH group or the OH end of the mRNA must be aligned with the active site of the RNA polymerase itself in order for the correct NTP to be added. And that RNA polymerase is also associated with the template strand of the DNA. So the misalignment will occur between the mRNA 3' OH group and the active site of the RNA polymerase. Now, how do you go about it? How do you recover? How does this RNA polymerase recover from this very process? So you need to, what it does is, suppose the RNA polymerase has been able to synthesize overdue or has been able to synthesize the mRNA beyond, and when it has encountered the blockage in the DNA, it was able to synthesize, so the RNA polymerase got stuck and some of the mRNA got synthesized. So there is that kind of misalignment. So you need to cleave some RNA, means some ribonucleotides from the 3' OH group, so that the 3' OH is realigned with the active site of the RNA polymerase itself in order for the reaction to begin again. So there are two steps here. Number one is pyrophosphorolytic editing. The pyrophosphorolytic and hydrolytic editing, this thing also comes under the RNA polymerase proofreading activity. Now, the pyrophosphorolytic editing does not really or does not always involve the grief factors. It just depends on the activity of the RNA polymerase itself to backtrack and dissociate the nucleotides and again realign itself with the active site of the RNA polymerase. Whereas in the hydrolytic editing, the E.coli uses GRI-A and GRI-B factors, the eukaryotes use the TF2S for the RNA polymerase 2 specifically, the transcription factor 2S. Now in specifically E. coli or in the prokaryotic system, GRI-A and GRI-B, these are the two specific proteins which help or which stimulate the cleavage activity and which assist in the cleavage of the overdue 3' OH, and it cleaves again backtracks and cleaves the whatever overstanding has been synthesized, and it will again realign the 3' OH group of mRNA with the RNA polymerase active site itself, and thereby the RNA polymerase can restart the synthesis of the mRNA. So RNA polymerase does contain these two types of proofreading activity. So just keep in mind. So this is about this is everything you need to comprehend the dynamic process of the elongation in the transcription in the prokaryotic system. So if you have been able to comprehend this video properly, then, and if you have any doubts, then kindly put your queries in the comment section below. And if you have liked the video, if you have liked the content, then don't hesitate to hit the like button, and also kindly subscribe to my channel, Rahul's Advanced Biology. Thanks a lot. See you soon.